Hi, my name is Nickellaus Roberts. I'm a PhD candidate at the University of Alabama, working with Dr. Kevin Kosot. And today I'm going to touch on some of the work that we have been doing sequencing multiple displacement amplification products for organisms that otherwise are too DNA limiting for doing whole genomics. When I talk about microscopic organisms, I just sort of want to ground everyone that we're working on microscopic metazoans. For this talk, I'm actually going to be focusing on Lepidodermella squamata. This is a gastritrick, otherwise known as the hairy bellies. If I were to actually show you the real size, I would actually have to take this circle and shrink it about 8,000 times. And that sort of gives you an idea of something being so microscopic. For our genomics projects, we really wanted to do HiFi sequencing, right? High fidelity sequencing, because the reads are long, they're greater than 15 kb, they're highly accurate, right? We're talking high levels of accuracy and reliability, and they're easy to assemble with a program such as HiFi ASM. We're really interested in low input sequencing, and particularly the ultra low input protocol from PacBio, because it allows sequencing even in the case of limiting DNA, right? However, it oftentimes, even though it is a 5 nanogram protocol, it requires oftentimes greater than 5 nanograms of DNA after shearing, size selection, library preparation, et cetera, and so forth. And so, for example, from a meiofaunal organism, or even for a model organisms such as C. elegans, right, we can only reliably obtain maybe 2.5 nanograms from a single organism. And if we want to avoid pooling to avoid heterogeneity, right, we would need to get much more from a single organism, and there's just not enough to do an ultra low input library from one animal. So we decided to focus on multiple displacement amplification. This is a single cell technology, and I'm just going to touch on sort of the gist of it real quick. So MDA, multiple displacement amplification, involves taking your DNA, adding random hexamers, right, those anneal. Phi29 polymerase is a viral polymerase. It will bind and begin polymerizing. It will knock off the 5' end that has annealed, right? The strand is displaced. More hexamers anneal, Phi29 begins to do its thing, and polymerization happens over again. If we just look at that last step one more time, right? But binding and beginning of polymerization, we can see that multiple strands are displaced, right? And we're getting lots and lots of replication of otherwise limiting amounts of DNA, right? So this is the multiple displacement part of MDA. Issues with MDA. MDA does have some amplification bias. It's been known to have amplification bias since its inception, and also chimeric junctions can form that can prove detrimental to assembly. The benefits of MDA, and it's particularly MDA in terms of other single cell technologies, it can produce 50 kb fragments from minimal material. We're talking micrograms from picograms, right? So this allows us to do regular input libraries for HiFi sequencing. It's also isothermal, it's fast, and it's cost effective. We decided we wanted to test this, of course, on some of our microscopic organisms. So we wanted to use Lepidodermella squamata. This is the one I mentioned earlier. And in order to validate this, right, we wanted to try this with half of a C. elegans. This is Bristol N2. We used QIAGEN's Repligy Advanced DNA Single Cell Kit, and then we did selective precipitation with the Circulomics Short Read Eliminator. And following some fragment analyzer QC, we felt confident in sending our fragments to PacBio, and we sequenced both of these organisms on a single PacBio Sequel II flow cell. I'm going to gloss over a lot of the bioinformatics in this talk. However, if you want to chat about it, we can talk about it after this. In order to assemble, we did HiFi ASM. We decontaminated using the blob toolkit workflow. We masked our repeats with repeat modeler and masker, and then we annotated with breaker. For C. elegans, we were really pleased with this result. So on the left here is the C. elegans HiFi only assembly. This is a de novo assembly of C. elegans from MDA amplified products. We get about 336 contigs and an N50 approaching a megabase. But most importantly, when comparing the BUSCO, that is the benchmark universal single copy ortholog score, with the C. elegans N2 strain, right, it's very comparable, right? And if we compare that to an assembly done as a reference guided assembly with ragtag, right, we're also getting really comparable results. And if we do a reference guided assembly, you can see we get an N50 close to that of the reference. The high number of contigs in the reference guided assembly is, again, just the reference guided assembly throwing sort of short contigs at the end there. We do still see amplification bias. So this is the fraction of the total read set versus the GC percentage of the assembly. This is a sliding 1000 kb window. So yes, we do see amplification bias. However, we were still able to get about 20x coverage total. And at our lowest, again, we're still getting 10x coverage. And this is still HiFi. So we're pretty pleased with that. It's comparable, again, to the reference in terms of BUSCO score. And it covers large portions of the reference genome with the drop-off in unexpected places, right? As an example, here's 4 chromosomes of the reference genome of C. elegans with our genome's largest contigs mapped against it. You can see that we're covering pretty much all of a particular chromosome, right? So we're getting good coverage of the reference for our de novo assembly from MDA amplified products. Most importantly, of course, is now that we can validate this approach, we wanted to see if it could work on a de novo genome assembly for a phylum of organisms, again, Gastrotreca, that did not have a genome, right? We did not know about genome size. We did not know about many aspects of this genome, and it was sort of a shot in the dark. And we can see that we were actually able to get a relatively contiguous genome. We get about 527 contigs and about a size of 140 megabases, right? And an N50 2.9 megabases, which we're very pleased with. You could see, again, looking at the fraction of the total reset compared to the GC percentage of the assembly, right? We're actually getting less amplification bias in this sample, which is good to see. As well, if you look at a camera plot, right, we're still capturing homozygous and heterozygous chimeras. Again, we were able to take something that otherwise wasn't tractable at all, right? So we couldn't do an ultra low input protocol. We would only be able to do short reads, right? Which can be still detrimental for assembly, right? If you've ever done a short read assembly, you know that it's very difficult, especially without a reference. And so we could take this, right? Something that normally we wouldn't be able to make a genome from and begin to ask and answer new questions, right? You'll notice here again that the BUSCO score is about 80.1%. To touch on that, right? So this is a pretty long branched organism, Lepidodermella squamata here, Gastrotreca. However, it's behaving really nicely in phylogenomic analyses, right? So we may not be getting chromosome level, but we're able to ask the questions we want to ask using this approach, right? And so we can mine out a lot of genes for phylogenomic analyses, and we can get much greater occupancy for this phylum than we normally would be able to do using transcriptomes. And it's behaving very nicely. So now that we have a genome, again, we get to do the cool genomics work, such as comparative genomics, right? We can look at orthogroup occupancy and content, right? We can look at comparing it to other published genomes from organisms that otherwise were easier to do with HiFi, right? Then they didn't require some finagling with MDA or ultra low input, but now we're able to compare them just on the same playing field. So, and to wrap it up, I'd just like to thank the members of the COSAT lab and my funding sources. This includes the Invertomics research project at the University of Oslo with Torsten Struck and Alberto Garcia Valero. I'd also really like to thank Michael Gilmore. This was an undergraduate in the COSAT lab who did a lot of the bioinformatic and lab work associated with this project. And yeah, thank you very much.